So we're in the week of October 5th through the 8th. So we finished articulation, so we're starting muscle tissue today. So I'm going to lecture for most of the class, and then we'll go over to the lab, look at the cadaver for a little bit. Midterm grades can be posted by October 12th, which is next Monday. We'll have a combination of your lecture and your lab grade. So just wanted you to make sure you're aware of that. All right, so for the module, so chapter 10 is actually in week 9. You have the lecture presentation, pre-test you can take. There's a PDF on classes of letters. So you have processes of muscle cells. So our properties of muscle cells, the full notes that are the YouTube playlist. So today we're going to start the lecture presentation. I also wanted to go over this properties of muscle cells because you will probably see some short answer question that asks you to list and describe a few properties of muscle cells. So we'll start here. So basically you would have to list the term and then give a definition. So one property of muscle cells is contractility. So it's the ability to contract where proteins in the cell get closer together. And it doesn't necessarily mean that the cell itself has to get shorter. All right, excitability is another property of muscle cells. So excitability just means that the cell can respond to a stimulus. Could be chemical, could be mechanical, could be electrical. Conductivity means the cell can conduct electrical charge. And it can go across the plasma membrane of that cell. The fourth property is extensibility. And that just means that the cell can be stretched without being ruptured. Okay? And elasticity, so the flip side of that is the cell can go back to its original length, okay, once it's been stretched. So extensibility is like being extended. Elasticity is essentially recoiling back to original length. So again, you need to know names and definitions, okay? Does that make sense? Okay, you need to know the names of each of those properties and give the definitions. All right, so let's start chapter 10. So in our study of tissues on histology, you learned about muscle tissue. You learned that there are 3 types: skeletal muscle, cardiac muscle, and smooth muscle, right? Everybody generally remember that, okay? So cells are specialized for contraction, okay? That's what they do. How do they move the body? Well, they pull on the bones. Muscles pull. Okay, even you think you're pushing, you're really still pulling, okay? Your muscles really aren't designed to push, they pull against the bones that they're attached to. So your skeletal muscle pulls on the bones. The cardiac and smooth muscles control movements inside your body. So we just talked about properties of muscle, like excitability, responding to stimulus, contractility, shortening, extensibility, stretching, and elasticity, recoiling. Functions of skeletal muscle include things like movement, maintaining your body position and your posture, supporting the soft tissues of your body, guarding the exits and entrances in your body, keeping your body temperature where it needs to be, and being a storage system for nutrients. Okay? So those are important properties of muscle. So the skeletal muscles, what do they have in them? Well, obviously they're going to have skeletal muscle tissue, right? They're skeletal muscle. They also have connective tissue. Also have blood vessels and also have nerves. So you got muscle tissue, you got nervous tissue, you have connective tissue, right? So you got lots of different tissues involved here. And remember, this is in Canvas, so if you don't catch something, you can go back either and watch the video or go back and pull the notes up, okay? So skeletal muscles have different layers of connective tissue. They have epimysium, paramysium, and endomysium. Okay, and we'll learn more about each of those. So in your epimysium, this layer of connective tissue is made up of collagen and it surrounds your whole muscle. And it's connected to what we call deep fascia, which is more connective tissue. And it forms a barrier to separate the muscle from tissues around it. The epi is sort of outer, okay? Remember that. Then we have paramysium, and anytime you see mice or myo, it's muscle, okay? So peri's sort of in the middle, and it surrounds fascicles, and these are bundles of muscle fibers, okay? So just like the epimysium surrounds the whole muscle, paramysium surrounds the fibers, the fiber bundles, the fascicles. Paramysium surrounds bundles or fascicles. So what is it composed of? Well, it has nerves, so you can stimulate it. It has blood vessels, so you can get nutrition, get rid of waste. It has elastic fibers and collagen fibers for structure. So in the endomysium, remember endo is the inner layer, and it surrounds individual muscle cells. And muscle cells are also known as muscle fibers. And what does the endomysium have in it? Well, it has networks of blood vessels, specifically capillaries. Okay, we'll get into cardiovascular system in AMP2, but they're small blood vessels. All right, myosatellite cells. These are stem cells which can divide rapidly, they're undifferentiated, and they can repair damage to the muscle. And you need that because anytime, if you think about those of you who do any kind of weight training or running or anything where you're stressing out your muscles, you're constantly in a state of damage and repair to grow. So you need that. You also have nerve fibers, so again, those nerve fibers can be stimulated. So the collagen fibers inside the epimysium, the endomysium, paramysium will come together, and they come together at the ends of different muscles. What do they make there? Could make a tendon, which is a bundle of those fibers. Or they could form an aponeurosis, which is a sheet. And in either case, the purpose is to attach skeletal muscle to some bone. Okay, they're attachment points. Because the bones can't move unless they're attached to a muscle. Because the bones give you structure, but they move because they're being moved by muscle. Okay, lots of words. So what does that actually look like? So we talked to this bionic gentleman who has a muscle outside of his skin. No, I'm just kidding. It's a drawing. All right, nobody laughed, but that's okay. All right. So you have the epimysium, which is on the outer layer. This is surrounding the whole muscle. Okay. So you see that surrounding the whole muscle. And then on the inside of that, you get this mass of tissue that surrounds these big bundles. Okay. So, and you can tell they're bundles because they have things inside of them, right? So, but each collection is a bundle. Okay, each collection is a fascicle. And those fascicles are surrounded by your paramysium. Okay. Now, inside each fascicle, you have fibers that are surrounded by endomysium, which we'll see soon. So here you have this cutaway. You have the whole muscle with epimysium around it. Then you have the kind of white space there, which is paramysium surrounding the fascicles. Okay, here's an individual fascicle, and inside it, you've got individual muscle fibers surrounded by endomysium. Okay, so this is that endomysium surrounding each muscle fiber. All right, but wait, there's more, all right? So here's your individual muscle fiber. So what does that look like in a cross section? Well, you've got individual myofibrils, okay? We'll learn more about those in a minute. You have your endomysium surrounding it. You've got capillaries, which will provide nutrition. And you've got mitochondria to make ATP, right? Muscles need lots of ATP, you need energy. Okay, here's your myosatellite cell, your stem cell. Okay. Here's your nucleus. Okay, you have multiple nuclei in skeletal muscle cells. Think about it. Why do you need multiple nuclei? What do nuclei do? Anybody remember that back in chapter 3? Okay, so yeah, so the nucleus houses what? DNA, right? DNA is in there. DNA is used to make what? Anybody remember? RNA. Remember transcription? You know, just kind of dust the cobwebs off a little bit, right? So you got transcription. That RNA leaves the nucleus, goes and finds a ribosome and makes protein in translation. Remember that story? Okay. Then also, what's in the nucleus? The nucleolus, right? Remember, the nucleolus makes ribosomal RNA, right? So you need ribosomes to translate that messenger RNA and make protein, okay? And you need lots of protein for muscles, right? Because they're building. You know, there's lots of proteins involved, as we'll learn in muscle contraction. So we're always building, okay? You know, try not to forget all that stuff that you learned a few weeks ago, because you'll see it again, okay? You'll also see it on the final exam. So don't just throw it all out, okay? Because if you don't forget it, it'll make the final exam a lot easier to study for. I can just tell you. All right. Organization of that skeletal muscle. So you have extensive vascular network. So what does that mean? Vascular means blood vessels. So you have lots of blood vessels to deliver oxygen, give you nutrients, take away waste. Okay. So when the skeletal muscles contract, they have to receive some stimulation from the nervous system. Okay. And skeletal muscles known to be voluntary. Why? Because you typically don't have to think to make your muscles move, right? You do have to think, rather, for most of them, right? So that would be weird. I mean, you get spasms occasionally, right? So sometimes you get a muscle spasm, muscle moves uncontrolled, but that would be weird if that happened all the time, right? Most of the time we think even though we don't necessarily think about what we're doing, you are consciously deciding to walk, to pick something up. to run, any of those things you're doing, you're thinking, you're making a choice to do it, okay? Your diaphragm typically is going to work subconsciously, okay? You do get some control, but generally it's going to work on its own, okay? Which is good because you don't want to be thinking to breathe, okay? Those of you who are singers maybe have a little more control over your diaphragm, but most of the time it's just working on its own. All right, so let's talk about the actual fibers in the skeletal muscle. So they are huge compared to other cells, right? They are extremely large. And that would make sense because they do a whole lot. And part of the reason they're so big is because they have multiple nuclei per cell. To the point of having hundreds of nuclei per cell. Okay, and we just talked about why that's so important, right? For making your RNA, making your ribosomes, okay? So extremely important. So how do they form? Well, we'll learn about that in the next slide, but basically you're fusing together myoblasts. And remember, anytime you see myo, it's muscle. Anytime you see blast, that means it's a developmental cell, it's a building cell, right? So these are early cells. Skeletal muscle fibers are known to be striated. They have lines you can see if you look at the slides, they're striated. So how do those skeletal muscle fibers form? Well, you have these embryonic cells, the myoblasts, and they start fusing together. And so if each of these has a nucleus and they start fusing together, you get one big cell with a bunch of nuclei, okay? So you end up with something like this when it matures, okay? Be up to 30 centimeters in length for one muscle cell. So if you think about, generally speaking, how small cells are in your body, and you think 30 centimeters is a length you can actually measure. So to think you have an actual cell in your body that's that long is pretty amazing. All right, so this gives you a really good view of the striations. You can see the lines, those small lines there. Okay, here's a picture, you can see that. There's all those nuclei. There's some mitochondria. Remember mitochondria, powerhouse of the cell, they make what for us? ATP, thank you. Make your energy. Those are all the myofibrils inside the skeletal muscle fiber. And we'll get into sarcolemma in just a minute. All right, so lemma is sort of a root word that basically means plasma membrane. Sarco is another root word that means muscle. So you'll see myo a lot, and you'll see sarco a lot. So this is a plasma membrane of a muscle fiber. So it surrounds the sarcoplasm of a muscle fiber. So, or cytoplasm of a muscle fiber. So sarcoplasm is cytoplasm. Sarcolemma is a plasma membrane. Okay, just in the muscle cells, they have special names. Okay, when we get to neurons, they'll have special names too. But it's the same thing you already studied. Okay, it's adapted for muscle tissue, but it's what you've already learned. So how do we get contractions? It means you change the membrane potential, okay? And again, we haven't studied a lot of that yet. We will. Basically, it's difference in charge between the two sides of your cell membrane, your sarcolemma. Okay? Another structure you need to know are the transverse tubules, or the T-tubules. And they are tubes, and they extend from the surface of the muscle fiber, okay? And they go deep down into the sarcoplasm, okay? So what do? They're going to transmit action potentials, okay? And action potentials basically are that change in electrical charge that allow for some process to take place, whether it's sending signals along a neuron or making a muscle contract. So those action potentials get transmitted from that cell membrane, sarcolemma, into the interior of the cell. So action potentials trigger contraction. Okay, so that's just kind of a big principle. Okay, no action potential, no contraction. Okay. All right, skeletal muscle fibers. You have the sarcoplasmic reticulum, which is, again, remember sarco is muscle, and plasmic reticulum sounds like endoplasmic reticulum, right? So this is basically the muscle cell's equivalent of the smooth endoplasmic reticulum. And it is tubular, so it's like a bunch of tubes, and it goes around the myofibrils. It also will form chambers along the ends of those T-tubules. Okay, so we can call them terminal cisternae because they're on the ends. All right, so terminal is the end. Cisternae, cisterns are essentially containers. Okay. Now, when you have a T-tubule on either side, you have terminal cisternae. That's 2 + 1 is 3, right? So we call that a triad. Okay, so sarcoplasmic reticulum, what's its basic function? Well, it is specialized to store and release calcium ions. And these calcium ions get transported from the cytosol into that terminal cisternae where they get stored and used when they're needed. Now, we think of calcium for bones, but calcium is extremely important for muscles, as we'll see. Okay, another term, myofibril. These are divisions in your muscle fibers. Okay, and that's where contractions happen. And believe it or not, there's another layer underneath that, or the smaller layer you have. So just like the muscle fibers are made up of myofibrils, the myofibrils are made up of myofilaments. You have two basic kinds of myofilaments. So they can be thin filaments or thick filaments. Thin filaments are made up of actin, thick filaments are made up of myosin. A couple of ways you can try to remember this. Actin is a thinner word, it's a smaller word. So it's a thin filament. Myosin's a bigger word, more letters. Okay, and it's a thick filament. Another way, you can say thin actin. Okay, so there's the the commercials. You probably haven't seen these because it was when I was a kid. So there's a foot spray called Tinactin, and they used to have these commercials. So they'll say tough actin, Tinactin, and I guess when I was trying to remember this, I would say tough actin, thin actin, and that would help me remember that the thin filaments were made up of actin. So whatever works for you, going by the number of letters or making up some kind of name, whatever works for you. So here's your muscle fiber, skeletal muscle fiber. Your sarcoplasm, okay, your muscle cell cytoplasm. You can see the striations, the lines. Here's all the nuclei. Okay, so your sarcolemma, plasma membrane. Your muscle fibers made up of myofibrils. Okay. Now let's look a little bit greater detail. Okay, so here's one myofibril made up of thin filaments and thick filaments. Okay, actin and myosin. All right, you can also see lots of mitochondria making lots of ATP. Here's your sarcoplasmic reticulum. It looks webby. Okay, don't get all bent out of shape about this. We'll get a chance to look at these in lab when we cover muscular system. Okay, you'll get a chance to look at models of skeletal muscle. But this is the sarcoplasmic reticulum. On the ends of the sarcoplasmic reticulum, you have the terminal cisternae. These are the T-tubules. If you're creative, you can see that you make this line going here and this line going right there. That makes kind of like a little T. Okay, so that's your T-tubule or transverse tubule. And if you combine one T-tubule and two terminal cisternae, you get a triad. Okay. And again, these are all myofibrils. It's your cytoplasm around that's your sarcoplasm, plasma membranes, your sarcolemma. Okay, a little greater detail, you can see again, myofibril, thin filament, thick filament, your actin, myosin, mitochondria, cell membrane, sarcolemma. So your muscle fiber is composed of myofibrils. And your myofibrils is composed of myofilaments. Okay, another view, again, the terminal cisternae, transverse tubules, terminal cisternae here, sarcoplasmic reticulum, two terminal cisternae, and a T-tubule is a triad. All right, so I'm going to stop there, kind of let you talk with your partner about the layers that you've learned about and the structures that you've learned about in there. So sarcomeres, these are within the muscle fiber, these are the smallest functional units. So here, your interactions between the thick and thin filaments create your muscle contractions. And the reason why you get this striated pattern in your myofibrils is because of the way that those thick and thin filaments are arranged. And you get this combination of dark bands and light bands. The dark bands are also known as A bands. The light bands are also known as I bands. Okay, how are you going to remember that? Well, what is the vowel in dark? A. So there's A bands. What is the vowel in light? I. Those are the I bands. Okay. All right. Learn about the A band. Okay. This is not a band on TV. It is a band in your muscle fibers. Okay. So, all right. The M line. It's in the middle or the center of your A band. And the proteins in the M line, okay, they are going to stabilize the positions of the thick filaments, okay? The H band is on either side of that M line, so just still in the center, but just a little bit past the M line. And there you'll see thick filaments, but not thin filaments, okay? Then you can see a zone of overlap, and it's dark. And it's dark because you have essentially a overlap of thin and thick filaments. Okay? So that's your M line, your H band, and your zone of overlap. For the I band, this is where your thin filaments are. So not the thick filaments, the thin filaments are there. Here you have, so in the A band, you had in the middle, you had an M line, right? On the I band in the middle, you have a Z line. And it's called a Z because it zigzags, okay? And it marks a boundary between sarcomeres. So basically from sarcomere to sarcomere is, or from Z line to Z line is a sarcomere, okay? From Z to Z is a sarcomere. Now you also have this protein called titin that's involved. It's very elastic and extends from the tips of the thick filaments over to the Z line of the thin filaments. So what does it do? Well, it keeps those filaments properly aligned, okay? Keeps them where they're supposed to be, okay? So they don't get off kilter. So they also help in bringing the sarcomere back to its original length, okay, once the sarcomere is relaxed. All right, so what does that look like? So here's a sarcomere. All right, so everybody say hi, sarcomere. All right, so Z line to Z line, that's your sarcomere. Okay, for your A band, this is your dark band. It's dark because of the thick filament. In the center of the A band is your M line. On either side of your M line is your H band. But you can see here, there's only thick filaments here, okay? And the one way I would look at it, like, how do you get H band from that? Turn your head sideways. Look right here. That. Okay? Doesn't that look kind of like an H? All right? So, because it would be confusing if you said, well, that looks like, you know, because this is the I band. So you got to turn your head and make it an H, okay? Because you don't want to have I bands in both places. That'd just be weird, right? So, all right, so this is your H band. So here's your zone of overlap, because you have thin filaments and thick filaments. Okay, here's your I band, your thin filaments, no thick filaments. Here's your Z line, zigzag, right? Zigzag, Z line. Now, what's this green thing over here? That's your titin. Okay, extends from the ends of the thick filaments to the Z line. Okay, and its role is just kind of help stabilize this thing, stabilize this sarcomere, keeps it all in place. And remember, myosin's the thick filament, actin's the thin filament. Okay, here's a microscope slide of it. Okay, here's your A band, your dark band, I band, your light band. Here's your Z line in the middle of the I band, M line in the middle of the A band. Space kind of on either side of the M line is the H band. Okay, and Z line to Z line is a sarcomere. Okay, so superficial view. Again, you can see the outside of the sarcomere. Okay, you can see your thick and thin filaments. This is superficial. So if you were then to do a cross section, it looked like this. Don't worry about this. You're not going to see any questions on this. We're just showing it to you. Okay. All right. We're just showing you position of some of these different filaments. Because you have to imagine, if this is my imaginary sarcomere, you know, if you look at the filaments from the side, it looks like this. But if you're looking at it from the inside, it would look kind of like that. Okay, so that's what those little dots are, basically. All right. So let's kind of summarize what we've learned so far. Okay. So what do we know? We know the whole skeletal muscle is surrounded by epimysium, right? And inside of that epimysium are fascicles. Now the fascicles are surrounded by perimysium, right? So this layer around, all of this bundle is perimysium, okay? And inside of the fascicle are muscle fibers, which are surrounded by endomysium, okay? Now that muscle fiber is made up of myofibrils, which are surrounded by sarcoplasmic reticulum, right? Those are our storage vessels for calcium. And what are they made of? Well, they are made up of sarcomeres. Remember our sarcomeres, Z line to Z line, made up of our thin and thick filaments, actin and myosin. Okay. Got your light band, dark band, I band, A band. And you also have your tighten that's helping stabilize it. Okay. Here's your M line in the middle of your A band, H band, where there's only thick filaments. All right, so this is it. You're looking at each slide individually. So again, your skeletal muscle is surrounded by epimysium, contains muscle fascicles, which is surrounded by perimysium. Contains your muscle fibers, which are surrounded by endomysium. And they contain myofibrils, which are surrounded by endoplasmic reticulum. They consist of sarcomeres. And again, here are your components of the sarcomeres. Okay, your I band, A band, thick filament, thin filaments, your titin, okay, your Z lines, your M line, and your H band. Okay. So before we move on to this, I'm going to put this slide back up and discuss with your partner levels of organization. All right. All right, so let's talk a little more about the filaments. So you have your thin filaments, and the thin filaments are composed of multiple components. You have F-actin or filamentous actin, you have nebulin, tropomyosin, and troponin. So your F-actin is composed of 2 rows of G-actin or globular actin. And the active sites, remember active sites, these are enzymes. There are proteins, some proteins are enzymes, will bind to myosin, which is the thick filament. Now nebulin holds the F-actin strands together. Okay, so you also have tropomyosin and troponin. So tropomyosin keeps the active sites on G-actin covered, and it prevents actin and myosin from interacting with each other. And the troponin, it will bind to the tropomyosin, the G-actin, and calcium ions. Okay. Okay, so here's what we already know about the sarcomere. And so if we take that and we kind of look in a greater magnification, what we'll see for the thin filament is that F-actin, which is those two G-actin molecules twisted around on each other. Okay, so that's your F-actin. Okay. Now, if we're going to look at even greater magnification, so what we'll end up seeing is tropomyosin, okay, which is essentially wound up as well, but it's kind of a long molecule. And then we'll see, and these are all G-actin. These are all those G-actin molecules. So the tropomyosin wraps around the actin. And it keeps essentially the active sites on the actin blocked. And troponin can bind to the tropomyosin and the G-actin. All right, so for the thick filaments can have around 300 myosin molecules per thick filament. The myosin molecule has a tail, and that tail binds other myosin molecules together and has a head. Okay, it projects toward the nearest thin filament. And I don't know if you remember this, but when we started talking about Titan, we said that it was very elastic. So it recoils after stretching, which is good because muscles do a lot of stretching. So here's your myosin, okay? So this is your myosin tail, this is your myosin head. And the head is attached to essentially what forms a hinge, and so allows that myosin head to cock back and forth and move back and forth. But these myosin molecules, will all have a head. All right, so now we're going to build on that. So important concept in muscle contraction is sliding filament theory. So this is one of those concepts that, we have short answer questions. So this is a potential short answer question of explaining the steps in sliding filament theory. Okay. So during contraction, remember the H band? Okay, remember the I band? So the H band was the area where you just had thick filaments. On the A band, your I band was the area of the thin filaments only. So they narrow. And these zones of overlap are going to get wider, get more overlap, which makes the Z lines get closer together. Okay, but the width of your A band remains constant. And you may say, how does that happen? Your myosin, your A band is composed of your thick filament. Your A band is composed of your thick filament. The molecule itself doesn't change length. Okay, so the width of your A band stays the same because the myosin molecule didn't actually get bigger or longer or shorter. Okay, so the width remains constant. but the thin filaments slide toward the center of the sarcomere, toward that inline, okay? So this is a sarcomere in the relaxed state. Okay, you have a large space here for your H band where there's only thick filaments. Okay. Now in a contraction, okay, so we look relatively, here we have a small zone of overlap. Okay, in a contraction, those thin filaments, those Z lines move closer together, then you get more overlap. You get a bigger zone of overlap, and you get a smaller H band as a result. Okay. So does everybody see how when you contract, your Z lines get closer together? Does that make sense? Okay, when you contract, does it make sense that you get more overlap of the thick and thin filaments? Does that make sense? Okay, if it doesn't, tell me. Does it make sense that your H band would get smaller because your overlap got bigger? Does that make sense? Now, does it then also make sense that your A band would stay the same size because the myosin molecule itself didn't change length? Okay, does that make sense if your A band stays the same because the molecule, because the A band is based on just on myosin, which didn't change. The only thing that changed was the amount of overlap. Okay, so if y'all understand that, y'all understand sliding filament theory, which is pretty good because it is not an easy concept to understand. Okay, so kudos to you. All right, so this tells you here that when the ends are free to move, ends of that contracting muscle fiber move toward the center of the muscle fiber. Okay, so when one end of the myofibril is fixed, the free end gets pulled toward the fixed end. So that should make sense. So if there's no attachment, it just sort of gets smaller toward the middle. But if there is an attachment point, everything slides toward the attachment. Okay. All right, so I'm going to let you all describe sliding filament theory to each other real quick. So I'll give you a couple minutes to do that. One here. So in your skeletal muscle fibers and your neurons, you have what we call excitable membranes. And you get what we call depolarization and repolarization. Okay. And that produces action potentials. Okay. And we'll get into, depolarization and repolarization later in the course. But they're basically phases of action potential. And action potentials are just electrical impulses. And so why do skeletal muscle fibers contract? Well, it's because of these excitable membranes inside of motor neurons, or inside of your muscles that get stimulated by motor neurons. So the motor neurons, they stimulate an excitable membrane in your muscle. Okay, it causes muscle to contract, causes depolarization, you get a contraction. Okay, another term, the neuromuscular junction. Okay, neuro, nerve, right? Muscular, muscle, junction is a meeting point. So don't get caught up by the word, just break it down. So it's a synapse between a neuron and a skeletal muscle fiber. Okay. And that synapse, I know we, at least in lab, we talked briefly about neurons a little bit when we talked about nervous tissue. All right, so they have an axon, and the axon terminal of that motor neuron, which is the axon terminal, is just the ending of that axon, release neurotransmitter, okay, into the synaptic cleft. And the synaptic cleft is basically that space between the axon terminal and the muscle fiber. And the acetylcholine, or ACH, is your neurotransmitter for muscle cells, or for those motor neurons. And ACH, or acetylcholine, will bind to sodium channels, and it opens them. And it's chemically gated. And these are sodium channels on the muscle fiber. So it'll go, essentially, if you have the axon terminal neurotransmitters, get released out through exocytosis. Remember that? So it goes out of the cell from those vesicles, goes across the synaptic cleft, attaches to a sodium channel. Sodium channel is open, allows sodium to go through. Okay. The sarcolemma. Sodium is going to enter the cell and cause a depolarization at what we call the motor end plate. And that generates an action potential. So we get electrical charge. All right, so what does that look like on the picture? So again, here's your vesicles inside of your axon terminals that have acetylcholine in them. Here's your synaptic cleft, space between the axon terminal and the motor end plate. Okay. So you get an action potential, then you get an electrical impulse, you get change in membrane potential, travels along the length of the axon, gets to the axon terminal, it sends a signal for those vesicles to go to the ending of the axon terminal through exocytosis, release their neurotransmitters, and they travel across the synaptic cleft. Those neurotransmitters are going to attach to those chemically gated sodium channels, and that's going to make sodium enter the muscle cell, okay, due to essentially facilitated diffusion, right? So it's forming essentially a protein channel. All right, so then, so once the sodium rushes in, you get an action potential, okay, you get depolarization. All right, so you get your action potential, then you have to have a way to get rid of the acetylcholine. So either acetylcholine can diffuse and go away from the synapse because the concentration is high here and low somewhere else, or it can be broken down by these structures here, acetylcholinesterase. Okay, and that allows that motor end plate to be stimulated again. Okay, because if you're just full of neurotransmitter, they're just going to stay open all the time. Those channels are going to stay open all the time. So there's some negative feedback there, okay? All right. So what I'd like for you to do is to go ahead and discuss with your partners the stages of what happens at the neuromuscular junction, okay? We'll pick up with excitation, contraction, coupling next time. We're not finished. What I'm going to have you do is, as you discuss, go ahead and get your stuff. We're going to go into the lab and we're going to look at the Syndaver, or look at the muscle system.